Today, we actually have Michelle King, and she's actually the manager of the USABO here at CEE. All right, Michelle, you want to take it away?
Thank you everyone for joining us at this TEP event. As Kim said, my name is Michelle King, and I am the program manager for the USA Biolympiad. I will plug that one of the benefits of the program, in addition to studying, you do get to explore the breadth of the biological sciences, but it's also preparing these students for intensive college courses that they may not encounter a lot of the topics, or these courses are very large, and so they can't get the help that they need. So this is a really good prep for those students. So today I was invited to just give a little talk about my dissertation work, which was on calcium signaling network and Pseudomonas aeruginosa. But I wanted to give you a little bit about my background and what actually got me interested. I fell in love with research, or I knew the moment that I wanted to do research when my biology teacher, who had a physics degree, kind of got a little tired of my questions. He wasn't tired, he couldn't answer them because he didn't have the background. So he pulled out one of his textbooks and he let me read it. And then my question wasn't answered, and he was like, well, if it's not there, we just don't know. And I was like, what do you do when there's information you don't know? Do we just, live with these gaps in our knowledge? And he was like, no. There are people who research stuff and they are on the front edge of knowledge, and they help us learn, advance, and put the information that's in your textbook. And for some reason, I had never thought about that before. I always just assumed that knowledge was there, and I don't know how it's accumulated, but I was learning it. And as soon as he told me that, I was like, wow, I could be the first person to discover something. And that's one of the things I carried with me through my research is I love so much that I am now the first person in the world to know this very thing, which is very exciting to me. So that was one of the draws. And then as soon as I saw a bacteria being streaked on a plate, I was sold. Microbiology was my passion. I knew I wanted to study microorganisms, and I wanted to study like how they interacted with the environment. I was very interested in biofilms. So I was very lucky as an undergraduate student to begin research in a lab studying calcium signaling in Pseudomonas. And I continued my work in that lab through a PhD, because not only was my research fascinating, but I had found a mentor who inspired me, motivated me, and encouraged me to follow my passion. And that passion ultimately led to STEM education. So here I am, the manager at the USA Biolympiad, and I'm so thrilled. But I am excited to share with you my work. So let's get started. So just the picture of this plate, this is just Pseudomonas growing on an agar plate. So the green color is one of the things that it's most known for. It was actually diagnosed by the green color. So if you ever have an infection that's green, most likely Pseudomonas aeruginosa, and also that may not be a good thing. Because Pseudomonas aeruginosa is a gram-negative organism. So you'll see it on the image on the left. So that's an image I actually took using our microscopy facilities at OSU. So it's ubiquitous in nature. It's found pretty much everywhere. But what I think is the most interesting part of this organism is that it's opportunistic, meaning that you encounter it on a day-to-day basis, but you will not get a Pseudomonas infection unless you're sick or unless there's an open wound. So one question that I always ask myself is, how do bacteria know when you're sick and when you're not? I mean, it's not like they're at the doctor's, they don't hear your temperature. So that was one of the driving questions of our research, actually. And Pseudomonas also causes both acute and chronic infections. It is so diverse in the infections it causes. Usually microorganisms will infect certain parts of the body, or they'll specialize, but Pseudomonas is just very talented and infecting everyone. And I'm apologizing because as a microbiologist, I love my organism, but I also hate my organism because unfortunately, in 2020, who characterized it as one, like the second most critical pathogen to study because of its antibiotic resistance. And unfortunately, Pseudomonas is resistant to pretty much everything on the market. So they use combinatorial treatments. And unfortunately, we are getting into a phase where those will no longer work. So we need to find other therapeutics and think about what Pseudomonas uses to adapt to the body. And the more we learn about this organism, the more we can manipulate it and save these people. So I know that this slide is going to be a lot, but Pseudomonas has a number of virulence factors. And virulence factors are just these factors that help it cause a really nasty infection. So at the top, you can see kind of this transition between acute and chronic. And so an acute infection is seen as like a very aggressive, very rapid, you will see something, you will get very sick in a few days. And then a chronic infection is prolonged. So Pseudomonas, so in one instance, which we'll talk about in cystic fibrosis patients, Pseudomonas can infect them for pretty much their entire lives and be perfectly happy in this environment. So it's very important to study the switch that goes on. And if you look under the slide, you'll see a whole bunch of information. But what it's showing you is that these are most of the networks that are characterized now that have been shown to cause this switch. And at the very bottom, you'll see that's basically the result of the virulence factors. So we have the type 3 secretion system, which is attributed to an acute infection, and it secretes toxins. You have secretion systems that secrete proteases, so proteins that degrade other proteases, lipases that degrade lipids, and then of course flagella, which the bacteria use to move, is very important in acute infection because they got to get around versus a chronic infection. They're a little bit more stable and stationary. And of course, something that fascinates me the most is the green color of Pseudomonas. And that is due to two very specific virulence factors, which one is a siderophore, and siderophores actually steal iron from the environment to bring back to Pseudomonas. And iron is required for life, and it's at such low levels in your body that there's a continuous battle between the organisms infecting you and your body to keep the iron present. And so siderophores are one of the mechanisms that bacteria can use to basically remove the iron from your body. And then of course, I would be remiss to speak about chronic and not talk about alginate, PEL, and PSL, and those are all polysaccharides, which help the bacteria get embedded into this like thick, sticky mucus. So it's very hard for antibiotics to penetrate. It helps keep nutrients in. It basically protects the bacteria until they're ready to break open and spread and then just keep growing. And Pseudomonas, as I said, is ubiquitous, and this isn't bad for us healthy people. Unfortunately for those, it is found, it's found in water systems. So there's some work to determine the best way to line the water systems to prevent biofilms from growing. So water systems like hospitals or in cities where hotels store some water, like on the roof, they've found biofilms there. The middle picture at the top is medical implants. So those are hip implants, which the biofilms will definitely reside. Hot tubs, one of the most known infections that it causes is hot tub rash. And that's a very, very mild and usually because of unclean hot tubs. And of course, the biggest thing is how it resides in hospitals. And so in the picture in the bottom left, every place there's a little indicator is a place that someone touches, and there's a potential to pick up Pseudomonas and spread it. And what's very concerning is that if you see in the middle picture, I have soap and I have ultrasound jelly because Pseudomonas has been isolated from soap dispensers in hospitals and other public places. And there were some instances where the actual, like the manufacturer of ultrasound jelly had some biofilms growing on one of their instruments. And so it actually contaminated a lot of this jelly. And so it wasn't as bad for like pregnant women, but for people where they use ultrasounds like for wounds and stuff, they were getting infected. So that's also a major problem. And then of course, when we talk about hospitals, we talk about how does it go from patient to patient? They're able to track them in wings, and they can track down to the nurse or the doctor who interacted with one patient or one reservoir, like the sink system shown in the bottom right, and then they go to connect with the patient, which of course, again, is very dangerous when we're in a hospital. So one of the primary environments that Pseudomonas is studied as a pathogen is within the lungs of the cystic fibrosis patient. And cystic fibrosis, for those of you who may not know, which a lot of people don't unless you have somebody close to you who suffers from this genetic disorder. So it's a genetic disorder in one of the chloride pumps in your cells. And this disorder, it causes a number of different things: mislocalization, degradation. It depends on the mutation. There's several different mutations characterized with cystic fibrosis. But the end result is an imbalance of ions. And so you can see with the second cell there, so it's talking about acidification and depletion of water. So the mucus in their lungs that is used as like a barrier becomes really thick and really heavy, and like ions aren't able to like freely move and be in balance. And so they're all kind of congested in these areas where the thick mucus is. And plus cystic fibrosis individuals, and there's a myriad of problems that they have due to these few mutations, but their cilia doesn't work. And cilia in your lungs are extremely important to help clear out this bacteria. So you have a lot going against you. And what happens is that at a certain point in time in their lives, Pseudomonas is introduced to a cystic fibrosis patient, and then their morbidity and mortality significantly increases. So there's much more damage to their lungs. There are other bacterial fungal pathogens that do infect the lungs, but it is been correlated with Pseudomonas infection that causes the most damage and leads to the most sharp decline. So the goal of anyone who's a parent of someone with cystic fibrosis is basically to prevent them from getting this bacteria introduced to them, because once it's introduced, it does not go away. As I said, with Pseudomonas, it can live in the lungs of a patient for 60 years. And ultimately, it is actually the cause of their deaths because they slowly suffocate. And one of the ways that my lab comes in is, of course, we have this question is like, how does Pseudomonas get into the lungs of a cystic fibrosis patient and be like, I'm in the lungs of a sick person? How does it do that? It's not, of course, it's a bacteria. And so we actually think that it's this imbalance in ions that actually tells the bacteria, oh, there's an imbalance, something is wrong, it's a really good time to infect. And my PI, as with everything in science, did a mild mistake in her postdoc, and she added extra calcium to the media that she was growing Pseudomonas strain in. And she noticed that, or no, she forgot to add calcium. And so she noticed there was no green pigment. It didn't grow. It wasn't, wasn't as healthy. It didn't do what it expected to do. And then she remembered she didn't add calcium. But that kind of told her that there was something in a relationship between pigmentation, which again, that pigment is like really nasty to humans. So there's some calcium regulation going on. And so our lab looked at the impact of calcium on these virulence factors. So to look at the motility and flagella, we did what was called a swarming assay, where you just inoculate the center of the plate, and basically the condition where they spread further induces swarming. So you can hear that between 0 millimolar, which is no calcium, and 10 millimolar calcium, there's not only an increase like diameter of the colony, but you see that green pigment. So there's definitely some calcium regulation. We looked at biofilm formation, and this basically the height quantifies how much biofilm there is. So there's more with 10 millimolar calcium. Those proteases I talked about at the beginning, they're more of them are made with extra calcium added. The pyoverdin, which is the siderophore I mentioned earlier, and another molecule, piocyanine, which I studied, those were both induced by calcium. And I just wanted to touch base on piocyanine because I worked on extracting it a lot as an undergraduate. And this molecule is redox active, which means it can enter a host cell and almost disrupt the electron transport chain. And it actually is very beneficial for the bacteria for a number of ways. So it's very important to look at what is regulating this as well. And then we performed virulence assays. And so there's a midrib, which is of a lettuce, which we injected. So PA01 is our like wild type strain. So 0 versus 5, you can see there's an increase in disease zone. And then we also injected Galeria melanella waxworm larvae. And you can see that in the 10 millimolar condition, there was more death. So it does actually increase virulence overall in addition to impacting these very specific factors. But why exactly is calcium so important? And I'm not sure like what your level of calcium signaling is, but until I entered this lab, I knew that calcium builds strong bones and then I needed to drink my milk. But I didn't actually know the role that calcium played in our cells. And this is a huge role. So our cells maintain a very low level of calcium. And then when it wants to do calcium signaling, it wants to signal your heart beating, it wants to signal apoptosis, it will increase the level of calcium and then decrease it. And that wave, that is a signal. And to our cells, they are so sensitive to the signal that the intensity, the amount of calcium that's in there, and how long that amount of calcium is in there will change how your cell responds, which is incredible. And our cells have a ton of different mechanisms to regulate this. So they have calcium channels, which allow calcium in. They have ATP-driven transporters, which will remove it from the cells. They have proteins that bind calcium and then either go perform a function or just bind calcium to remove it from the cytosol. And then of course they also have calcium stores because you need some place to store all that calcium so you can increase it and decrease it rapidly. So that is like the ER. The mitochondria does have some calcium, but more so the ER has been shown to be the major calcium stores for cells. And in your heart, the circa, which is like a specialized ER, is also a storage for calcium. And that helps regulate your heartbeat. Calcium regulates almost everything. I encourage you to Google just videos where they visualize calcium signaling. I was watching one earlier around a wound, and you can see the calcium waves around the wound, which is signaling to the cells to start growing. When we are fertilized, when our egg is fertilized, it's actually a calcium wave around the embryo that starts life. So it's just, so calcium is incredibly important for us, but when my PI started her research, nobody had thought that bacteria could be capable of this type of regulation. Oh, sorry, yes. So we have all this data and we showed that calcium impacts all of these, external things basically. And this cartoon kind of represents like what we knew. So we knew that bacteria actually have calcium transporters that are similar to the transporters in our cells. We know that bacteria have calcium binding proteins that are similar to the calcium binding proteins in our cell. And what's interesting is those calcium binding proteins are actually involved in signaling and not just removing the calcium. So there's got to be some kind of signaling component here in bacteria that we're not missing. And in an environment like cystic fibrosis, calcium is elevated. So this elevated calcium could be one of the major triggers of Pseudomonas that says that you are sick. And so we also know one very important thing, and this was shown in both E. coli and Pseudomonas. And if you look at the bottom where we have this little wave, so we measured intracellular calcium of Pseudomonas aeruginosa. And what we see is the point of the challenge is the bottom, and then there's a sharp increase, and then it goes back down to basal level. So bacteria also have this intracellular calcium regulation, potentially. Of course, we don't, and we never anticipate it to be as complex, but if you're living in environments where calcium is present and that's an important signal, it's important that we learn about it, especially for Pseudomonas. So our first goal was to look to see how Pseudomonas responds to calcium. So bacteria, to respond to extracellular signals, they have two component systems. The first component is a sensor that will bind to something, and the next component is a regulator, and that regulator goes and binds DNA. And so that's how they're able to regulate gene transcription. So our first question was, is there a two-component system that's going to be regulating this increase in these virulence factors in response to calcium? So in collaboration, we performed what's called a microarray analysis. It's where you can obtain all the mRNA in the cell and you wash it over this neat little chip, and you basically get intensities, and you can compare conditions. So we basically saw in the presence of calcium, Pseudomonas has over 62 component systems, and there was one that was upregulated in the presence of calcium. So we're like, this has got to be it, because two component systems, they're self-feeding, so the signal that activates them should be probably the signal they respond to. So we named it CAR-SR for calcium regulated sensor, calcium regulated regulator. And the next thing that our collaborator did was he made strains that did not have the regulator. So there's no way that the bacteria is able to respond to calcium. And there were two genes out of the entire genome of 5,000 plus genes, two genes were no longer regulated, and that is CAR-O and CAR-P. Yeah, so they were no longer regulated, meaning that these are the direct targets of this two-component system in the presence of calcium. And so my work studied CAR-P because we used a tool called iTassr to predict the three-dimensional structure. We actually predicted several calcium binding regions, which were very exciting to us because of course we want to look at calcium binding proteins when we're looking at the calcium signaling network. And it is predicted to form a 5-bladed beta propeller, and beta propellers have an incredible array of functions in bacteria and eukaryotes, which are very exciting. So we are very excited to follow this. And then one of the next things you do when you're trying to discover what a protein does is you look at its functional domains. And we can do this through comparing lots of sequences. So if one sequence is characterized to remove a phosphate group, if that sequence is found in 8 million proteins, then those 8 million proteins probably all do the same thing. Unfortunately for me, we didn't find anything that was actually helpful for the functional part. Just gave us some more questions. So it was exciting just to follow up on those. And in collaboration with one of the several undergraduate students that I worked with, we wanted to look at how conserved it was. Is it only found in Pseudomonas aeruginosa, which is like the main pathogenic strain of the Pseudomonads, or is it found in all Pseudomonads, including environmental strains that are non-pathogenic, because that can tell us like what role it's playing. So we looked at the currently available 2197 Pseudomonas aeruginosa, like all of the available Pseudomonads. We only found this gene in Pseudomonas aeruginosa. And there were actually 3 other species named, but we did an analysis to compare the genomes between Pseudomonas aeruginosa and these other, and they were misidentified as protogens otitis and mendocina. So those were all recharacterized as Pseudomonas aeruginosa. And the Pseudomonas SPs are strains that they haven't characterized down to a species level. And we actually, using CAR-P and this other analysis, we were able to verify that they are actually Pseudomonas aeruginosa. And this held true when we looked at all bacteria. There were a few samples that showed CAR-P was present and it was like a staph strain or something strange. But when we looked into the data, that specific sample was contaminated. So this is telling us something really exciting, that it is not shared with eukaryotes like some of our other proteins. It is not shared with other bacteria like some of our other proteins. This is very specific to Pseudomonas aeruginosa. So the next part of his project and another project with another student that I worked with is we actually proposed the hypothesis that this can be used as a biomarker to identify Pseudomonas aeruginosa in clinical samples. Because right now, depending on your hospital, how much money they have, what techniques they use, most hospitals don't honestly identify infections, which as a microbiologist makes me very upset. But if a hospital can do a simple PCR and have a simple PCR machine, and this can be done by high school students, then you can actually test the isolates, and within 3 hours, 4 hours, if you got to count running a gel, you can actually tell if there's Pseudomonas present or not, where in the past it would take 2 to 3 days, or there would be very expensive techniques. And so this is an opportunity for like researchers and doctors to use CAR-P as a biomarker. But again, it tells us something extremely interesting, is that this protein is very specific for Pseudomonas, and it must be very important. So what we wanted to do, I'm gonna just admit I was working towards protein work. I wanted to crystallize it. As with all science, it did not go smoothly. So we kind of we went in a lot of different directions to just uncover as much as we could about this organism. And I'm sorry, I'm gonna speed through this slide. But what we really also wanted to know is, how specific is it to this calcium signaling network? We've actually seen that it plays a role in swarming motility. So when you have a mutant that doesn't have CAR-P, there's no induction of swarming, so there's no increased diameter, there's no pigment. So we were wondering, if it plays a really important role, why would it just respond to one cue? And so we decided to perform a promoter activity assay, which is, we utilize something that bacteria and organisms use, which is the lux operon. And so the lux operon produces light. If you're interested in that, I encourage you to read some about Vibrio fischeri and the relationship it has with jellyfish, because they use this light as part of it's very interesting. So we use the lux operon, and we can put it in a plasmid. So we have a circular piece of DNA, and upstream of that lux operon, we put the DNA region upstream of CAR-P. So that region upstream of the gene, as you guys all know, should be where everything is regulated. And so basically we can add a bunch of stuff and we will see if there's a reaction, because there's more light, which is actually really cool. And I just kind of wanted to walk you through these techniques, because I worked a lot with them over my past 3 years. So the most important thing for microbiology work going forward and doing any real experiment is normalization, because we have to kind of synchronize our cells. And that's actually really critical, because if it's done wrong, your entire experiment that you've spent 2 days preparing for is done. And so we normalize it. We give it fresh media with and without calcium, and then we put it in this really awesome instrument called a plate reader, or a Biotech is the actual brand that we use. And it's able to not only measure the light coming from the well, but it can also measure how much bacteria is present. So what we can do is we can normalize it, because you don't want when you look at the graph at the end, you don't want the increased luminescence just because of the increase in cells. You want to see that even with an increase in cells, you're still getting an increase in luminescence. So this graph just kind of shows you what the kind of data we'll be looking at. And so you can see at 6 hours, which is mid-log, there is a sharp increase in the light produced and then a decrease. And so that's a piece of data I will be talking about in just a second. So of course, our first question was like, does it respond to calcium? And if it responds to calcium, how much calcium? We know that a very high concentration of calcium induces CARP, but there's subtle gradients everywhere. And so we grew different promoter constructs and increasing levels of calcium. And as you can see by the graphs, when we add calcium, the tops of the peaks increase, and they do kind of shift too, which is really interesting. So that's saying that yes, calcium is increasing the production of CARP, but different concentrations of calcium may do it differently. So that tells us there may be multiple levels of regulation. And then we put it in a strain that doesn't have that transcriptional regulator, CAR-R, no calcium induction, which is what we anticipated. And we did what's called a calcium challenge, which was a very fun experiment. It's where I took it out at the middle of its growth or the very end of its growth, and I added calcium. And as you can see, the responses are significant and they're completely different. And that's important because at mid-log, it's in the middle of the logarithmic phase. That's where all the bacteria are growing and getting really fast and getting all the nutrients. And then stationary phase is where they're running out of those nutrients and they stop growing as fast and they kind of start to die off. So the fact that there are these differences is telling us again that there are other cues. And in terms of like so if you think back to acute and chronic, so when there's low amounts of bacteria, it's kind of acute. Maybe CARP is being regulated differently in these infections versus when Pseudomonas switches to a chronic infection and there's a lot of bacteria. So that was also very interesting. And this response isn't present without CAR-R. So that is the two-component system that is regulating this gene. We also looked at another important ion, which I told you was iron. So this is also imbalanced in the CF lung. And so we tested it originally as basically a way to prove reviewers wrong because they told us that calcium is not that important. There's like magnesium and iron and blah, blah, blah. We tested magnesium and zinc, no response, not special. Then we tested iron and we did get a response. And then our next question was, in these environments of the lungs, it's not just one ion or the other, it's both. You're going to be exposed to both elevated iron and elevated calcium. And our original thought was there would be no difference. It would just increase and that's it. But you can see from the green line, it increases and it keeps going and it goes until the end of the read. And we don't know when it stops, but this is just a huge activation that we did not anticipate in the combinations of these ions. So what we think is like, maybe these ions do positively regulate it, but there's other things about the environment that will kind of help temper this increase. Or CARP is just really important when there's elevated calcium and elevated iron. And then this response is not seen in the CAR-R mutant. So that two-component system is not just responding to calcium, it's also responding to iron, which is incredibly interesting. And next we wanted to look at, again, more of the role that it plays in the body. So we looked at the role of the response to the mutant in response to hydrogen peroxide. So our body uses hydrogen peroxide and other reactive oxygen species to kill off the bacteria. That's one of the major immune mechanisms. And you can see when we expose our cells with added calcium that the mutant that has no CARP, that's indicated with the little triangle, has less survival. So it's impaired somehow in this response. And again, this is all painting a picture that it is important for infections. And so we looked at its role in the Galeria melanella waxworm, which you can see in the top right-hand corner. And the elevated calcium is indicated by the black. So you can see that the mutant that has a little triangle in front of it is the percent survival is a lot lower actually than the wild type. And then when we put the gene back into the strain, it's restored. So this is telling us that there is some kind of balance. We know it plays a role in survival. It's extremely conserved, so it must be very important. But then there are these like subtle differences that don't really agree. And so there may be something larger here that we don't see because we cannot study in a human. That is illegal. And this is a little rough slide, but I wanted to throw this in because I also think it was really interesting. So we quantified the mRNA of CAR-P in our lab strain and also 2 cystic fibrosis isolates. So that is the 31314A and the 22014B. And I'm going to just say that I would like to thank the Oklahoma Department of Health and the individuals who contributed these sputum samples, because it takes a lot to contribute sputum samples, and they do it for the betterment of our research, and we really appreciate it. And so you can see when looking at the transcription in these isolates, so yes, in CAR-P, it's elevated in response to calcium. Already know that. But in the isolates, what's interesting is that the first one has a significant response. So it's very significant compared to wild type. Second one has less of a response, which is that speaks more to the heterogeneity within this environment and between strains within the cystic fibrosis patient. Because if you plate out Pseudomonas from a CF patient, you don't get one type of colony. You get multiple colonies. You get small colonies that will grow in 4 or 5 days because they're so slow growing. And then you get these colonies that grow immediately. And if you turn the plate upside down, the biofilm is so thick, it just oozes. So there's all this heterogeneity. And so we think that this is actually, we're going to follow this up and see what types of isolates these are to see if that could maybe explain the differences in response. Although all 3 strains showed an increase of CAR-P in response to calcium, which again follows our hypothesis. So just to sum it up, I unfortunately graduated before we could really determine the role of CAR-P in the cell. But I was fortunate to work with a number of other projects. So this image kind of shows you what we know about calcium signaling in Pseudomonas. So we know that CAR-SR responds to both calcium and iron. We don't know if it binds directly, but that's something a future study. And we know that this regulation regulates CAR-P. And then CAR-P is also regulated by oxidative stress and intracellular calcium, which we did not end up talking about because I helped characterize a putative calcium channel, which is actually responsible for generating that peak and bringing all the calcium in, so making the initial signal. So the CAR-P transcription was low in this mutant. So intracellular calcium is playing a role somehow. So we know that it's regulated by a lot of these factors that are very important for infections. It is found in every Pseudomonas aeruginosa that has ever been sequenced. It's highly conserved. It's very novel just to this bacteria, but we don't know what it does. And fortunately, I have a whole team of people still working on it back in grad school, and we got to publish a few more papers. So all this work has been published in combination with undergraduate students that I worked with, and of course my wonderful PI and other collaborators. So hopefully one day we will know what CAR-P does, but I'm going to tell you when we find out, it's going to be really, really, really important. And so with that, as with any researcher, you would be remiss to thank the teams of people that helped you. I had an incredible set of collaborators, and my mentor to me is my female science icon. When I told her that I wanted to leave research because I thought there are bigger problems right now in STEM education, bigger than antibiotic resistance, bigger than these pathogens, bigger than the unknowns, is the lack of STEM education and the lack of appreciation for microbiology in high schools. And I'm like, I see this as a bigger problem that needs to be resolved, and I want to help. And so when I told her I wanted to leave the lab, I was very emotional, and she was like, you are passionate, you follow your dreams, and she supported me the entire way, and that's not normal. So I had the exception. And then of course I did have the privilege of mentoring a high school student. So you can see her accepting award in one of the bottom pictures. And then I can't underscore the value of having a good mentor and good lab team, and just you become a family, and I'll love all these people the rest of my life. And with that, I will take any questions.
All right, so one question is, like, what do other propeller proteins do?
That's a really good question. So as I said, they're really diverse. Not only so they go from like 5-bladed beta propellers to 9-bladed beta propellers, and they can do everything like degrade sugars, degrade lipids. Sometimes they'll just bind to things and then perform a function, but a lot is in degradation. And then of course we looked at some beta propeller phytases, and that's actually where we're leading towards. So phytases remove phosphate from phytic acid, which doesn't mean a whole lot unless you know what phytic acid is. And phytic acid is actually how, like, plants in our body store ions and inorganic phosphate, which is incredibly important. And so phytases actually remove the phosphates and remove the ions, and so that the bacteria can utilize them. So we're thinking that carpi has a role with phosphate and calcium.
Awesome. And so you mentioned like that carpi is really conserved and also very unique to Pseudomonas, but that calcium signaling is not. So what are other sort of, I don't know, I guess conserved proteins maybe? I'm not sure what the, like between prokaryotes and eukaryotes, like what are like some big ones?
Yeah. Okay, so I'm just, of course, talking calcium signaling because that's what I know. So there's one very specific protein domain. It's called an EF hand domain, and this hand coordinates calcium. And this domain is found in one of the most important signaling proteins in my mind in our cells, which is calmodulin. Calmodulin has 4 EF hand domains, and then calmodulin, once it binds calcium, it goes and binds other partners, and then that induces a major signaling event. So EF hands are found throughout eukaryotes and prokaryotes. Pretty much every branch of life has EF hand proteins. And so we identified one in Pseudomonas. The trick is to identify what it binds to, because we know that it's binding calcium, and that because it has this hand, that it's involved in some kind of signaling, but we don't know what the signaling or binding partner, and that's what was being investigated when I left. And then the calcium channel that I mentioned is actually very similar to a calcium channel that is in the ER that is regulated by pH. But interestingly enough, our protein doesn't have the residues that would make it respond to pH. So it's responding to something different, which is exciting.
What do you think, what's your best theory for what carpi does?
Oh, I mean, I've thrown out a lot of theories, like hypotheses for the function of carpi. Like I said, there is a lot of relationship between calcium and phosphate in bacteria. In E. coli, phosphate and some other molecules form channels that allow calcium in. In some other organisms, phosphate's used to store calcium. So bacteria don't really have a place to store except potentially the periplasm. So phosphate could be something. And then carpi could be degrading these to allow calcium in and regulating its function. It could also just buffer calcium, because our hypothesis is that while bacteria don't have organelles, the periplasm is actually how they store calcium for signaling events in the future. So carpi could be doing something related to that.
Oh, interesting. And then just as a, just so everyone knows, she's actually sitting across the hall from me right now. But I didn't know if you wanted to talk about anything with USABO for a couple of minutes, or share anything. It's
Yes,
exciting.
I definitely can. I'm incredibly excited. As I said, like moving towards STEM outreach was my passion. I don't see enough kids interested in science because science is cool. I mean, it's fun. When I would work in Oklahoma with kids, they would have this very negative outlook on microbiology, and they didn't want to study science because it wasn't for cool kids, and it just like really opened my eyes to like how we need to shift the communication in the topic. And so when I saw the USA Biolympiad, like although it's a competition, and that's plain and simple, it is a competition. We want our team Biolympiad USA to go represent our country, you know, in the IBO and like win gold medals as with every other Olympiad. But what I love most about it is we provide students and teachers a way to start science clubs, start USABO clubs. We have resources that you can use to teach your students, but we also really want students to take the lead. And we have some clubs where you see students thrive. I have students this year that were emailing me and they had all of the information for their school and they're like, this is our proctor, but I'm in charge of the club, so I'm going to organize it. And I mean, it's just a joy working with these students who they feel so confident because they have these skills and they've been working and developing. And I also think with these clubs, it gives students the ability to teach other students. And the best way we learn is to teach others. So it's a really great experience. And it also brings together people who love science and just want to talk and learn and explore. There's still not enough microbiology in the Biolympiad for me. Actually, one of the topics for IBO is microbiology and biotechnology, so that's pretty exciting. But it's just so exciting to see how into it these kids get. I mean, if I had the opportunity to really, I mean, I came from a small school in Oklahoma. There are not many people, you know, primarily white. Just I had science teachers who were teaching on a field. You know, my classes were rather large because of budget cuts. We are like, Oklahoma's not known for state funding for science and education. And so just thinking about the impact that this would have made if I really had something to like give me the drive to study. And if I had been exposed to these topics a lot earlier, and of course it's all on me, I could have, you know, done Google searching, whatever. But if I had been able to like really explore and look at the material, like I've looked through the materials that we have in the USABO, and I like protein data bank, you can go and you can look at molecules of the month, and there's nothing more beautiful than a 3-dimensional protein structure to me. You know, there's all these cool resources that I think would have really helped me not only just like explore the passion and explore the field, but also like prepare me for college. Because when I entered into my intro to bio course and there was 350 people and my TAs were not bio TAs, they were TAs from other departments who just needed placement. And, you know, I was like, the topics were interesting, but it was hard for me to just follow and keep up at the fast pace. Because I loved my biology teacher, but he's a physics teacher. And, you know, he wasn't necessarily always on topic and, you know, X, Y, and Z. But it would have like knowing those concepts a lot earlier and at least hearing those words, I think would have like made me more confident in what I knew and like helped me absorb better. Because if you learn something at first when you enter university courses, you need to study 8 million times before you really absorb it. But if you hear it in high school and you're like already casually reading about it, and then you do some studying for this and you're in a club and you're like constantly talking, when you get to your intro to bio course and they're talking about population dynamics, you're like, oh yeah, I know exactly what happens to plants once volcanoes come through. I know exactly all, you know, plant anatomy. I got all this. It would make my freshman experience, which is jarring, going to college, first time you move out, first time you have complete freedom, it would have made it a bit easier. And so I really love the opportunity to give these students like, hey, study, have fun, take an exam, just one, just see what it is. And then you really get to explore. And then our exams are written, they are not go to your textbook, read a definition, and verbatim repeat the definition. Our exams are designed to get students to think about how scientists see their data. So based on what you know, what do you think is going to happen? Or what technique do you think you should use? So it's really trying to push kids into that mindset of like, you're doing research. What would you see? What do you see from these results? Data interpretation. So that's something that I really loved coming from a high school of fill in the blank worksheets and just very, matching. And that, I mean, that was all it, I'm not, I think those are very valid teaching tools, but when you have teachers who teach out field and that's all they have to work on, and then most of your science teachers are coaches and history teachers are coaches, I kind of felt like, I got left out of it. So I would have loved to have this opportunity, and I'm so excited to like be able to provide this opportunity for students.
Awesome.
And, yeah.